Hi, my name is Brooke Jewell. I'm a Winthrop University McNair Scholar, and my project is entitled Temperature Optimization, Expression, and Purification of the Bacteriophage-Derived Protein Cain-55. And this work was done under the mentorship of Dr. Jason Hurlburt. So before I get into what a bacteriophage is, I want to go over why this research is important. So as you probably know, antibiotic resistance is a huge problem in the world today. The CDC estimates that 2.8 million antibiotic resistant infections occur each year. And of those 2.8 million antibiotic resistant infections, 35,000 deaths occur. So here on the right, I included a figure which describes how antibiotic resistance can occur. So due to natural errors in DNA replication, you can end up with an antibiotic resistant mutant bacteria. So of course, this represents the bacteria population within a human. So let's say this person gets sick and gets prescribed antibiotics. The antibiotics will kill all of the bacteria that are not resistant, except for that resistant mutant, and then that resistant mutant will proceed to take over the population, leading to this person being more resistant to the antibiotics that they took. So how is this relevant? Bacteriophages in recent case studies have been shown to be potentially an alternate to antibiotics, so this is extremely important information. So let's go over what is a bacteriophage. A bacteriophage is essentially a virus that uses mycobacterium as a host for reproduction and survival. Cain belongs to the Syphilviridae family. Typically, mycobacteriophages belong to the Syphilviridae or Myroviridae family. Cain has 102 genes, 30 have no assigned function, and 4 have been identified to be cytotoxic. So this is our experimental overview, sort of all the checkpoints we went through in our research. So of course, Cain-55 is a gene within a bacteriophage that codes for a protein of interest. So we start with the target gene, and then to get more background information on our protein, we are going to generate homology models. So we can use web servers such as Fire2 and RaptorX to predict the structure and function of our protein based on proteins that we have already discovered and crystallized. So then once we have that background information, we're going to clone into target plasmids. Then we're going to transform E. coli with the plasmids, and we're going to put our plasmids into E. coli. Then we're going to grow our recombinant cultures. We're going to induce protein expression and harvest by centrifugation. We're also inducing with IPTG at 1 millimolar concentration. We're going to lyse the frozen cell pellet and purify recombinant protein using methods such as MCAC. Then we are going to attempt to characterize and crystallize the purified protein. And as Dr. Hurlburt says, fame and glory, now get back to work and start a new project. So as I said, we're going to start with homology modeling. So this is Fire2. Like I said, it is meant to predict structure and function of a protein. So when you submit the amino acid sequence for Cain-55, the closest match is acetylbiohydrolase. Now we have a couple of reasons to believe that this is a false lead. First of all, the confidence score on here is 48.5%, and the coverage score is 23%. So these are pretty low scores. Also, bacteria do not synthesize cellulose, and acetylbiohydrolases are meant to break down cellulose. So realistically, we do not believe Cain-55 is a acetylbiohydrolase, and this is one of the main points of our hypothesis. So like I said, we want to possibly disprove that Cain-55 is a acetylbiohydrolase. It has also been shown that Cain-55 binds to NUS-A, which is a transcription regulator. So if we can understand Cain-55 structure, we will be able to really get into depth on how it interacts with NUS-A and thus how Cain-55 regulates bacterial transcription. So the first sort of pre-experiment we're going to do here is temperature optimization of protein expression. The whole purpose of this first experiment is to test which strain and temperature is best for producing Cain-55. So we grew up cultures and grew them at 18 degrees Celsius, 25 degrees Celsius, and 37 degrees Celsius. We also had a median antibiotic control. just to make sure that there's no contamination within the media or antibiotic. So like I said, we grow up our cultures which contain Liria bertani medium, antibiotic, and glycerol stock. So we had PET 21 culture and PET 21 strain and PET 28 strain. For the PET 21 strain, we used ampicillin as an antibiotic, and for the PET 28 strain, we used kanamycin. So we grew up these cultures, and then when they were ready to induce, we induced half of them, one from each set, and then we continued to grow them at their separate incubation temperatures. We grew them overnight, and then the next day we spun them down, discarded the supernatant, and we were left with a pellet, which we then lysed using beeper and rapid boil ice cycles. Then we spun that down one more time and collected the supernatant, since the pellet will contain heavy things such as cell wall fragments and other things that we're not interested in because we want the protein. So then we used, like I said, that supernatant to perform SDS-PAGE, which is gel electrophoresis for proteins. So these are the results from our experiment. As you can see, each gel represents the different strain. From these results, we concluded that 18 degrees Celsius was the best temperature for producing cane 55 by recombinant E. coli. Also, PET 21 is the more favorable strain. Cane 55's molecular weight is 33 kilodaltons. So these boxes represent where we would see cane 55. Next, we go into purification. So for purification, we used MCAC, which stands for metal chelation affinity chromatography. Cane 55 has a his tag, and since histidines have a high affinity for metals, when we run cane 55 through a nickel column, the histidine will bind to the nickel. The next step, or one of the later steps in MCAC, is elution, which is when the sample is washed with a high concentration of imidazole. And when that high concentration of imidazole comes in, it's going to kick off the his tag, leaving a more purified protein. So here's our graph from the MCAC. The blue line here represents sample application, so when we actually ran our sample through the column, and the green line here represents elution. So you can see shortly after the elution stage, the his tag is kicked off the protein, which produces this peak here. And so we are going to use the fractions under here, or coordinates, to create yet another gel. So as you can see here, here's the gel that we ran after MCAC. So the reason we ran this gel is to see if our protein is actually fully purified, because actually cane 55 is pretty acidic, so purifying it is going to be a little difficult. So we ran this, and as you can see here, cane 55 is not purified because there are other protein up here and also a little bit below. So we're going to have to go through some other methods in order to purify cane 55. We also ran a Western blot in order to test identity. So like I said, the gel is going to test purity, and Western blot will test for identity. Since cane 55 has a molecular weight of 33 kilodaltons, we can know that this is cane 55. Also, at a higher molecular weight, we can see that cane 55 is interacting with something else. We don't know what it is. Like I said, cane 55 is shown to bind to NUS-A, so it could be interacting with DNA or RNA. We don't know. And down here, we can also see that cane 55 is breaking down, since it is at a lower molecular weight. So here are my references and also my acknowledgements. So Dr. Jason Hurlbert, who is my mentor, Dr. Frost, Dr. Fortner, who is the director of the Winthrop McNair program, Barb Yeager, the McNair Scholars Program, and also my lab mates, Priya Patel, Eric Walters, and Kyla Bonikowski, and of course the Winthrop University McNair Scholars Cohort of 2022. So thank you for listening. I'd be happy to take any questions at my email here, jewellb2@mailbox.winthrop.edu. Thank you.